In case of type 2 hypersensitivity, cell destruction occurs. That is the sign of type 2 hypersensitivity. And this cell destruction is mediated by antibody. And this antibody is very specific, like IgG and IgM antibody. Okay. So these two antibodies responsible for type 2 hypersensitivity. In case of type 1, I have mentioned IgE antibody is responsible, IgE. Now, this antibody can destroy this cell by three mechanisms. One is complement activation and the formation of a complex called membrane attack complex. The second is opsonization, phagocytosis, and the third is antibody dependent cell mediated cytotoxicity. So this is known as ADCC. And the example of this type 2 hypersensitivity is, the first is transfusion reaction. The second example, this is the example of type 2 hypersensitivity. The second is hemolytic disease of newborn. And the third is hemolytic anemia. The first is transfusion reactions. Transfusion means blood donations. And as we know, there are four blood groups: A, B, AB and O. What is the basis of this blood group? There is antigen on red blood cell. In case of blood group A, the RBC have a antigen. This is also known as agglutinogen. B, for B blood group, B antigen is present. For AB blood group, A and B, both these antigen are present. For O blood group, there is H antigen. So let me draw a RBC. Let's assume this is a RBC. Okay. And this RBC have antigens like this. Let's assume this is the H antigen. This is A antigen. And this is B antigen. So on RBC, there could be any three. At first, I want to mention here the antigen is not protein in nature, it is carbohydrate in nature. So this antigen actually carbohydrate. Three sugar moiety are actually compose the H antigen. The first is galactose, the third is galactose, and the middle is N-acetyl glucosamine. And these three molecules actually represent H antigen. So those who are O blood group, they have these three sugar moiety on their RBC, only this antigen. Then if it is A blood group, then what happens? In this galactose, another sugar is added. As I have mentioned this red color, so I am showing this sugar molecule as a red. So they have another sugar molecule that is attached with this H antigen, that is called N-acetyl galactosamine. Then he is A blood group. Okay. Now, if any individual have another galactose sugar moiety, then it is B group. So now you can see this H antigen is the base. Now by enzymatic reactions, here could be a galactose or N-acetyl galactosamine, and this A and B antigen are produced. Okay. Now I will show you what happens, why this transfusion reaction occurs. Antibody are present in our serum that actually cause the problem. Very interesting thing occur. Individual is blood group A, they have anti-B antibody. Those individual is blood group B, they have anti-A antibody. Those who have AB both antigen or AB blood group, they do not have any antibody, and those who have O blood group, they have anti-A and anti-B antibody. From our childhood, we remember, we memorize this point. But have you questioned that why this blood group A individual have anti-B antibody? Because we know, those who understand immunology, they know that if there is any antigen in our body, in that response of that antigen, antibody develop. I want to mean that if I have a antigen or any antigen coming from the outside, then I will develop a antibody against that antigen. Now you can see here the blood group A individual have A antigen, but this individual have anti-B antibody, against a antibody that is against the B antigen. So how this individual is actually getting the B antigen? Because until or unless our B cell can experience any antigen, it cannot generate a antibody. So you can see here that the blood group A individual have blood group A antigen. Okay, so this individual will not have anti-A antibody. This is very simple because self-tolerance. I have told you in my previous class that there is a self-tolerance. Our body immune cells are generated or mature or developed in such a way that they do not recognize the self antigen. So a blood group A that have a antigen will not generate anti-A antibody. That is very simple for self-tolerance. The reason is self-tolerance. But how this anti-B antibody is coming? But we memorize this fact. This B individual have B antigen but have anti-A antibody. So this B group of individual do not have A antigen. So how this anti-A antibody is coming? So if you know this fact or if you have any idea about this problem that I have mentioned here, you can write me in the comment section. I will give my answer later. Accidentally, if this individual donate blood to the B blood group individual, then what happens? This anti-B antibody will react on this B antigen. It will react with this antigen and complement activation occur. And reverse is also true. That is, if any B individual donate blood from B to A, what happens? This B group individual have anti-A antibody that will bind with the anti-A antigen and actually activate this complement pathway. As the AB blood group don't have any antibody, so it can receive both A and B blood because it have no antibody, so it will not react. But in case of O blood group individual, they both have anti-A anti-B antibody. So if we transfuse A blood group A blood to the O, then what happens? This individual O blood group will react with this A antigen and also will react with the B antigen. So hemolysis occur and the RBC will be destroyed. So this is called type II hypersensitivity. Now I will show you hemolytic disease of newborn, that is erythroblastosis fetalis. For this, Rh antigen is responsible. There is another antigen present on RBC that is called Rh. So let's assume if the mother is Rh negative, this is the conditions, and if she is fertilized by a father that is Rh positive, then always the child will be Rh positive. So if it is the first pregnancy, during the delivery the placenta and the uterine cell were actually separated. So during that time some fetal umbilical blood cord cells can enter, RBC can enter into the mother's circulations during the separation or delivery. This mother, Rh negative mother, have some B cells that can recognize the Rh antigen. So there is some B cell that can recognize the Rh antigen, and in response of this Rh antigen, this B cell will produce memory B cell. This memory B cell is capable to secrete IgG antibody that can cross the placenta. So what happens if the mother and father gets another baby, second time pregnancy, this mother have memory B cells against the Rh antigen, and during the second time pregnancy, memory B cell is producing IgG antibody that can cross the placenta, because IgG antibody can cross the placenta, and when it enter this IgG, when it enter into the fetus, this IgG antibody actually activate complement pathway, formation of MAP occur, and the RBC will be destroyed in the fetus, so the second baby will not survive. But nowadays almost entirely this event can be prevented, because scientists have discovered a antibody that is called ROGAM. So this antibody is injected into the mother, and this antibody actually binds with the fetal antigen, so that the mother IgG antibody cannot bind with the fetal RBC antigen. Another treatment is available, that is called plasmapheresis. The mother plasma is separated or discarded actually. So this is hemolytic disease of newborn, that is called erythroblastosis fetalis. Now I will talk about the third condition, that is hemolytic anemia. And the reason behind this is some drug like penicillin, cephalosporin, streptomycin. These are the antibiotic, and very well known medicine like ibuprofen can also cause this hemolytic anemia. What happens here? This medicine or drug, these are the drug, are adsorbed on RBC and bind with some antigen. Our B cell can recognize this drug and antigen complex and make antibody against this complex. And also these three processes can occur: complement activations, opsonization and ADCC. So by this way the RBC cells are destroyed and anemia occurs. This is hemolytic anemia due to this drug